Good morning friends, today we will talk about the life cycle of an angiosperm. As we know that angiosperm is the largest division of plant kingdom, we will see that how is the life cycle with the help of a cyclic presentation here. We will see the male gametophyte is known as your anther. Anther is in angiosperm flower there are two sex organ, one is male sex organ and second is female sex organ. In the male sex organ, as we can see, that is known as stamen. Stamen has main part that is called as anther. Anther is bilobed and which is joined with the help of connective and it have one filament. So, anther is a bilobed joined with connective, it has filament. When you cut the anther, you will find the tetrad inside the anther in the case of angiospermic flower. So, here we have taken a Brassica compressed that is called as mustard plant. Flower we have taken mustard plant and the flower which have the male reproductive organ as well female reproductive organ. In the male reproductive organ we are talking about anther, anther which is bilobe joined with the connective and it has filament. When you see cut the section of the bilobed anther, you will see this type of sporangium. This sporangium is known as microsporangium. This sporangium is known as microsporangium. Here microspores are present in that and they have the chromosome number which is called as X or N. The chromosome number N means to say that it is haploid. Now, here we will see that this is female part of the angiosperm which is known as megasporangium, and here you will find in the case of megasporangium when we cut the LS section we will see this. This is called as the stigma, the upper part where the anther have a pollen grain which used to come and land on it, and then you will see the long tube like structure which is known as the style, and this is a swollen part which is called as an ovary. So, from ovary you will see when we cut the section we will see the megasporangium which is also known as ovule. So, in this ovule section we can see the megaspore which is and also known as megaspore mother cell and the chromosome number is haploid. So, we can represent it by N. So, what we can see here in the case of anther microsporangium which have the N chromosome, and here we can see a lot of pollen grain. When we enlarge the microspore or the pollen grain, they are like this, they are haploid in structure. This enlarged diagram is showing here. When it get mature, the pollen grain mature, we will find that this outer layer which is a zigzag layer, it is also called as exine layer, and here the inner layer which is also known as entine layer. Now, we will see that inside this is called as microspore pollen grain. So, this is a pollen grain cell which is haploid in nature. Now, from this the exine dissolve with the help of a chemical produced by female gametophyte or by the unfavorable condition, and the formation of pollen tube take place. So, here the formation of a tube formation taken place, and these are the gametes which are called as the male gametes here inside the male gametophyte. So, this is a diagram of a male gametophyte where the pollen grain develop, exine will dissolve and the entine will develop a tube like structure, and these cells are known as your gametophyte and they are X in number or N in number which is known as haploid. Now, in the case of female part in the flower, we will see that this is the structure of a female part. Here the stigma is shown. When we cut the LS section, the upper part is called a stigma which is giving a landing platform to the pollen grain, and this long tube is known as the style and swollen part is known as ovary. So, ovary consists of ovule and they have the polar nuclei. So, 4 polar nuclei at the upside and downside inside the ovule, and which is enclosed with the help of integuments, and upper end is known as chalazial end and downside end is known as micropylar end. What happen in this? One cell will migrate from upward to downward and one cell will migrate from downward to upward. So, there are two type of cells which is known here, it is found. One is known as X synergid and second is known as egg apparatus. So, when the both haploid cell will fuse at the center, it is called as secondary nucleus. It is called as secondary nucleus. and the secondary nucleus have a chromosome number, that is called as your 2n, which is diploid in nature. But what happen in this, you will find when the pollen tube will come and fuse, it will make again it will fuse from secondary nucleus, it will make a triploid. So inside this, the endosperm having a chromosome number, that is a triploid in nature. So but when we talk in this, the all cells which is found in female gametophyte is haploid. So from ovary, when it discharge, we will see the megaspore mother cell which having the haploid chromosome number. As we can see this in large diagram, this is egg apparatus, as well as a downside, this is called as egg synergids, and both the one cell will move to the center of the ovule and they fuses there, this is called egg synergids. So here the diploid structure which is called as secondary nucleus having the 2x chromosome. In large diagram of this is shown here, and this is the female part of the Brassica compresstis flower. Now what happened, this N chromosome which is coming is haploid male gametophyte, it will fuses with the female gametophyte here which is also haploid, then N will added or fuses with the N female gametophyte and they will make what, zygote, then the zygote formation will take place. So the zygote formation, they have the diploid cell, means chromosome number will be 2n, and this condition is known as sporophytic or 2n generation, diploid generation. And here as we are seeing that the male gametophyte and female gametophyte, this condition is known as gametophytic haploid generation. So we are now at the position of saprophytic, sporophytic 2n generation, means to say diploid generation. So from the zygote, the formation of embryo take place. So from the zygote, the formation of embryo take place. So here the formation of coleoptile as well as coleorhiza take place, and from coleoptile the formation of plumule, and from plumule the formation of shoot take place in angiosperm, and in the case of coleorhiza the formation of root take place, right. So from root, the primary root, secondary root, tertiary root, there are two type of roots which can be seen, that is tap root and adventitious root. But in the case of angiosperm, when we see, there is only adventitious root is found, tap root is found, where there should be a primary root and then formation of secondary and tertiary root will take place in the case of when the embryo will develop. Now we will see the second in large diagram. After the development, the formation of this plant twig take place. This is a mustard or plant twig, or you can say Brassica compresstis plant twig, which is showing the sporophytic stage. So from sporophytic stage, this is the root region, as I have already told you, tap root is seen here, and dicot leaf as you can see here, which have proper reticulate venation, and here you will see the flower as well as pod carrying the, especially those mustard grains which used to give oil also, it can be used for various purposes, or seed formation take place, and from the flower only we can see again the anther as well as the Megasporangium development take place, as we can understand. So they used to make again gametophytic haploid generation. After when we see the flower is developed, it will make, when they get mature, they will make gametophyte generation which is haploid in nature, and after both the male and female gametophyte, when they combine together, they will make sporophytic diploid generation. So by this way we will understand the angiospermic cycle, life cycle of an angiosperm. We will also understand that angiosperm is divided into two parts. One is known as your dicot and second is known as monocot. So dicot, there is 75% of the plant is dicot which is seen nearby, and 25% of the plant which is seen in the particular environment are known as monocot. So in dicot they have special functions, they have two cotyledon, in monocot they have one cotyledon. In monocot the parallel venation is seen in leaf, but in the dicot the reticulate venation is seen. In the case of monocot, when we see, basically they have adventitious root, but in dicot there is a tap root. So by this way we can clearly define that angiosperm is the advanced form of the plant kingdom, which is dividing into monocot and dicot, and this diagram showing the life cycle of an angiosperm. Thank you.